Wow. Do I have to answer? Cloning my first gene. Who do I want to piss off more? My finger. This finger's been dislocated. I've been uncool my whole life. Joined the Unitarian choir with how hard it is to grow food, but then when it does grow, I have a list of about 5. I love looking at cells. I would say that. I knew I had found this gene. I guess the older I get, just the more grateful I am that I get to explore. The world outside of the cell that's equally important to within the cell is trying and failing all the time. I'm Dave Sherwood. I'm professor at Duke University in the biology department. I've been there 21 years. And I got my graduate degree there. Working in sea urchin development, and then I went out to Caltech and joined the lab of Paul Sternberg and fell in love with C. elegans and studied cell invasion through basement membranes, and then I got into basement membranes through my work on cell invasion. Oh my God, do I have a hobby? So many hobbies. I love basketball. My finger, this finger's been dislocated so many times, that's as straight as it goes. I love singing. I recently joined the Unitarian choir, which I love. I just discovered yoga, and I absolutely love that. I'm revisiting my love of acting, so I joined an acting group. Many hobbies. Gardening. Oh, yeah, gardening is wonderful. It's so humbling how hard it is to grow food, but then when it does grow, it's just so good. Yeah. Blackberries. Fresh blackberries are the absolute best. You know, I don't. I love looking at cells. I would say that, and but I don't have. I mean, I started off graduate school looking at notch signaling, and it was such a fascinating molecule in that it signals by having the intracellular domain cut and going into the nucleus. But I soon realized, with a cell, C. elegans genome encodes 20,000 genes, our genome about 20,000. It's really hard to have a favorite when they all work together. So yeah, no, not really. Wow, that's a great question. I would say I have a list of about 5, really. And like my first was a dominant negative to notch signaling in the sea urchin embryo, and it was incredible. The arc enteron invaginates and all the mesoderm comes off. And with a dominant negative, it was just a smooth arc enteron. I knocked out all the mesoderm. It was just marvelous. Cloning my first gene in C. elegans was amazing. It was evil 5. It's kind of funny, everted bulba 5. And I remember when I found the cosmid that rescued, all of a sudden, rather than being sterile, all the worms were fertile, and I knew I had found this gene. I would say when I was studying cell invasion, and we were wondering, does this go through a basement membrane? And I found an antibody that worked to laminin, and I saw this cell invasion event in C. elegans where this little hole was cut out of the basement membrane. That was incredible. And more recently, I find just wonder and value out of just about every project, right? We just discovered a new organelle in C. elegans that comes off of the nucleus, and it looks like a rapid way that the nucleus signals to the cytoplasm by dumping out a bunch of nucleoplasm in a rapid manner. And just, I guess the older I get, just the more grateful I am that I get to explore. And all of these discoveries are just magical. My most common method is visualization. Oh my God. I mean, it's like looking up at the stars, looking at cells. And so my lab has 3 very expensive spinning disc confocals, and the discoveries they've made is just amazing. Yeah. Well, getting into cell invasion, and basically it was from Wadsworth's group. They had tagged laminin, and we got their strain from them, and it just was beautiful, right? All of the different basement membranes throughout the worm, and then when genome editing hit C. elegans and Dan Dickinson and in Bob Goldstein's lab had figured out a high throughput method. And then I had a crazy graduate student that wanted to tag all of the basement membrane components. And I'm like, why not? Let's do it. And we did it. We tagged most of them, and it was a multi-year effort. And just the things on those confocals that we saw, the rapid movement of basement membrane components, like a slip and slide within the matrix, the different levels, the different functions. It was just this world outside of the cell that's equally important to within the cell, but it's been neglected. I mean, I was sold. It's like this. We understand a lot of the rules inside of the cell, the kinases, the molecular switches, the control mechanisms. What controls what's outside of the cell, right? How does the cell, and then what's that language between the cell and the matrix? It's this ancient, beautiful language that we barely understand. And what a gift to explore that. Oh, I'll go before that. As an undergraduate, I went to Illinois Wesleyan University, and I had Bruce Criley was the head of the department, and just an inspiring figure, as was Bob Hippensteel. He was a physiologist, and and Bob in particular, just really thought deeply about questions, and I love that. And they gave me the confidence that I belonged, which is so important. And then I went to Duke University thinking I'd be a physiologist, but instead I ended up in a developmental biology lab, Dave McClay's lab. And Dave just was also filled with this sense of wonder and belief, and he got so excited when you made a discovery, and that just translated into confidence. And I loved the sea urchin community. It was very small, and so you go to sea urchin meetings and you just feel like you're important. And that was such a great incubator for me. And then I joined Paul Sternberg's lab, and Paul is just this unique, crazy human being who thinks non-linearly, and I'm very linear. And I remember meeting Paul, and he was just all over the place. And when a problem was not like we'd expected, he'd be like, "Great, it means it's more interesting." And that kind of embracing of complexity, embracing of non-linearity, embracing of creativity was just incredible in my own career. And then I would say my last mentor is the marine biology lab at Woods Hole. So the MBL, I've been going up there for 23 summers, surrounded by 24 students from around the world in the embryology class, and every one of them has been a mentor, because every summer I rediscover the joy of discovery and of wonder and of people coming together in a common purpose of learning and discovery. I think that what I love about the matrix biology field, first and foremost, is how humble, unassuming, genuine, kind, dedicated. So once again, I remember my first Gordon Research Conference. Jeff Miner invited me, and I gave a talk, and it went over so well. And I just had this gratitude for Jeff finding me out of nowhere and inviting me, right? And it built from there. And then my next conference was a basement membrane meeting in Manchester, and I met Billy Hudson and all of the Manchester community, Ambra and Roy, and I just felt welcomed. And Peter Yurchenko. Peter said, I remember my talk, "What you've just described makes absolutely no sense and contradicts everything I've ever learned, and I love it." And how can you not love a community like that embraces things when you turn things on their head? And so, yeah. And then Rachel, my God, and Andrea. Andrea Page McCaw is just such a clear thinker, and that's so rigorous and boils things down to their essence. And then Rachel and I have known each other for, 10, 15 years, and she inspires me just about every day, because we now collaborate, just with her can-do attitude, her positivity, and her ambition. So many mentors. And you, Sergey, you do great work. My God, you inspire me. I would say to be positive, and be growth-oriented, and to really explore. I think that when you're young, what you should be doing is trying and failing all the time. So you want to do a combination of figure out what you love doing and figure out what you're good at. You want to put those two together, right? You know, I figured out pretty early on, I love basketball, but I wasn't good enough at it, right? So instead it became a hobby. And through really a combination of, I was so lucky as a kid to have parents that took me backpacking, telling stories. My mother was an English teacher, so I became a good writer early on. And that's been so wonderful in my career. And I found out pretty early that biology came easy to me, and writing about it, telling stories and writing papers came easy. So it was and then I loved teaching. So, you know, I fell into this career because I loved it, and I was good at a couple of the core components. And so I would say that too, if you're a good writer, but you don't like bench science, man, there's really a need for good writers. If you're a really good bench scientist, but don't really like writing and writing grants, you know, you may want to explore being in industry rather than being a PI. I've been uncool my whole life. I don't know if I can tell anything cool. I would say that the thing that impresses me about this society is the same thing as the basement membrane community. It's just how unassuming and welcoming they are. And I would say the other thing is that, my God, the people that run it, the Matrix Biology Journal, are just to their core incredibly well run. And I don't know if that's cool, but it's genuine and solid. And also Kendra, who's our administrator, you could not ask for a finer human being, more dedicated, more positive, kind of being the backbone that and the glue that connects us all together. That's not cool, but it's real and it's genuine and it's solid, and I just really love being part of ASMB. Oh, dogs, my God, that was an easy one. Why'd you ever ask that? Oh, now that's a harder one. Wow, do I have to answer? Who do I want to piss off more? You know, I would say laminin because we tagged that first in C. elegans. Okay, but a very soft preference.